we're going to be studying DNA structure, replication, and organization in detail. First, describe the experiment that contributed to establish DNA as the hereditary molecule. DNA is your deoxyribonucleic acid. So the first experiment was done by Griffith. He was a bacteriologist and embryologist in 1928 who was working on the pneumonia vaccine. Two strains of bacteria he used was Streptococcus pneumoniae. This bacteria gives pneumonia in human, and it is a bacteria that mice can defend itself against. So he took living S strain of this bacteria, living R strain, dead S strain, and he mixed living R strain with dead S strain, and he got the following results. Let's look at the Griffith experiment. The ability of S strain to induce disease was due to its ability to produce a capsular polysaccharide coating. This outer polysaccharide coat enabled the bacteria to conceal itself from being detected by the host immune system, which otherwise would get targeted and killed. Griffith injected mice with living R strain and heat killed S strain and expected that mice to remain healthy. But the mouse developed pneumonia instead. He then isolated the bacteria from the blood of infected mice from which S strain of bacteria were isolated. This implied that there must be genetic material in the heat-killed S strain of bacteria and a mechanism to transfer this material from dead S strain of bacteria to living R strain, which as a result changed the non-virulent R cells to virulent S cells. So for the Griffith experiment, must remember he took living virulent S strain, mouse dies. He took living R strain, which was non-virulent, mouse lived. Dead S strain heated, mouse lived. And for a mix of living R strain and dead S strain, mouse again dies. So these were the method used and the results. Next experiment was done by Oswald Avery and Colin MacLeod and McCarthy in 1944. It was eliminated by the discovery of DNA's role in transformation process. Only then it was concluded that DNA was a material that transferred from one cell to other, eventually changing R strain into the S strain. In their experiment, for the first time, researchers isolated almost pure DNA from S cell and mixed this with extracted DNA with culture of R cells. They observed that the transformation of R cell into S and thus confirmed the role of DNA in genetic transformation. Furthermore, they discovered the transformation ability was not altered by destroying protein by using proteinase and RNA by using RNase in this DNA isolate, but this ability was completely lost when DNA was destroyed. This experiment by the trio researchers proved that DNA to be a genetic material. Then we have Hershey and Chase experiment. The experiment began with culturing of virus into two types of medium. One set of viruses, A, was cultured in the medium of radioactive phosphorus, whereas another set, B, was cultured in the medium of a radioactive sulfur. They observed that the first set of virus A consisted of radioactive DNA but not radioactive protein. This is because DNA is a phosphorus-based compound while protein is not. The later set of virus B consisted of radioactive protein but not radioactive DNA. The host for the infection was E. coli bacteria. The virus were allowed to infect bacteria by removing the viral coats through the several blending and centrifugation. Observation of Hershey and Chase was E. coli bacteria which were infected by radioactive DNA virus A were radioactive, but the ones that were infected by radioactive protein viruses B were non-radioactive. They concluded that resultant radioactive and non-radioactive bacteria infer that the virus that had radioactive DNA transferred their DNA to bacteria, but viruses that had radioactive protein did not transfer to the bacteria. Hence, DNA is proved to be a genetic material while protein is not. Next is outline of the structure of DNA molecule. DNA molecule in 1953 was discovered by Watson and Crick. The work of Rosalind Franklin led to Watson and Crick's discovery. Franklin first has pointed out that DNA is made up of two spiral. Structure of DNA we know is a double helix structure because it looks like a twisted ladder. The sides of the ladder are made of alternating sugar, deoxyribose and phosphate molecules, while the steps of the ladders are made up of a pair of nitrogenous base. These four types of nitrogenous bases are adenine, thymine, guanine and cytosine DNA pairing. The nitrogen bases have a specific pairing patterns. Adenine always pairs with thymine and guanine always pairs with cytosine. This pairing pattern occurs because the amount of adenine equals to the amount of thymine, as I said, and amount of guanine equals the amount of cytosine. The pair are held together by the hydrogen bonds. DNA is a double-stranded helix, that is, each DNA molecule is comprised of two bipolymer strands coiling around each other to form a double helix structure. These two DNA strands are called polynucleotide as they are made up of simpler monomer units called nucleotide. Each strand has a 5 end with a phosphate group and 3 end with a hydroxyl group. We say it as each strand has a 5' end with phosphate group and 3' end with hydroxyl group. The strands are anti-parallel, meaning that one strand runs in a 5' to 3' direction while the other strand runs in 3' to 5' direction. The two strands are held together by the hydrogen bonds that are complementary to each other. Basically, DNA is composed of deoxyribonucleotides. Deoxyribonucleotides are linked together by 3' to 5' phosphodiester bonds. The nitrogenous base that compose the deoxyribonucleotides include ATCG, adenine, thymine, cytosine, and guanine. The complementary of strands are due to the nature of nitrogenous base. The base adenine always interact with thymine on the opposite strand via two hydrogen bonds, and cytosine always interacts with guanine via three hydrogen bonds on the opposite strand. The shape of a helix is stabilized by hydrogen bonding and hydrophobic interactions between bases. The diameter of double helix is 2 nanometer and the double helical structure repeats at an interval of 3.4 nanometer, which corresponds to 10 base pairing. Here you can see the structures of adenine, guanine, thymine and cytosine base pairs. We know that adenine and guanine are purines in nature and thymine and cytosine are pyrimidine in nature. Major and minor grooves of DNA. On the diagram you can see this is the major groove of DNA and here is the minor groove of DNA. As a result of double helical nature of DNA, the molecule has two asymmetric grooves. One groove is smaller than the other. This asymmetry is a result of geometrical configuration of bonds between phosphate, sugar and base groups that forces the base group to attach to 120-degree angles instead of 180 degrees. The larger groove is called major groove, occurs when the backbone are far apart, while the smaller one is called minor grooves and it occurs when they are close together. Since the major and minor groove exposes the edge of base, the grooves can be used to tell the base sequence of a specific DNA molecule. The possibility for such recognition is critical since protein must be able to recognize specific DNA sequences on which to bind for the proper function of the body and cell to be carried out. Then we have major enzymes and proteins of DNA replication. Number one is helicase. These are special unwinding enzymes that help in breaking the weak hydrogen bond which hold the two strands together. Topoisomerases, these are the enzymes that can break and release one strand of DNA. Then we have primase. These enzymes help in formation of primer. Primer is a short RNA segment that is formed on a DNA template before the replication can begin, which is essential. It polymerizes the complementary RNA building block A, U, G, and C, which stand for adenine, uracil, guanine, and cytosine in the primer. DNA polymerase. This enzyme is main enzyme required for DNA replication. It can link free DNA nucleotide to form the complementary strand of DNA. It polymerizes nucleotide in 5' to 3' direction only. So it is only in one direction. It is also known as a DNA dependent enzyme as it uses a DNA template for polymerization of deoxynucleotides. DNA ligase is an enzyme that can join the short section of newly synthesized polynucleotide chain. It puts the short pieces together after replacing the RNA primer with DNA. Repair enzymes. These enzymes can cut off the wrong bases and replace them with the correct ones. They help in rectifying the error that may have occurred during the replication. Let's look at steps involved in DNA replication. Number one is initiation. In prokaryotes, DNA replication takes place in cytoplasm, whereas in eukaryotes, it takes place in nucleus during the S phase of the cell cycle. During initiation, the DNA is made accessible to proteins and enzymes that are involved in the replication process. There are several specific chromosomal locations called initiation point or origin of replication, which is known as ori, from where the replication of DNA begins. There are no specific sequence for origin of replication, but there are certain proteins that recognize and bind to it and allow other proteins necessary for DNA replication to bind at the same region. DNA replication is semi-discontinuous in eukaryotes. Bacterial and viral DNA has a single origin of replication. It functions as a single replication until called a replicon. But in eukaryotic DNA, there are several origins of replication or replicon, so they are multi-repliconic. In the absence of ori, replication will not occur. We have elongation of DNA replication. This is the second step in DNA replication. Elongation starts with activation of deoxyribonucleotide. Deoxyribonucleotides occur freely inside the nucleoplasm in form of monophosphatase. So they are the first phosphorylated and changed to active form of the triphosphate with help of the enzyme phosphorylase along with energy. Number two, exposure of parent's DNA strand. Enzyme helicase act over the ori site and unwind the two strands of DNA by destroying the hydrogen bond. It results in formation of replication fork or Y fork. Due to unwinding, supercoiling gets developed on the end of the DNA opposite to the replication fork, which is released by the enzyme topoisomerase. Formation of RNA primer at the free 3' end of the one strand and fork end of the second strand. A small strand of RNA called RNA primer is synthesized with the help of enzyme. RNA polymerase or primase RNA primer function like the 5' end of the new strand to be synthesized. Base pairing, the two separated DNA strand in the area of replication fork now function as their nitrogenous base attracts complementary phosphorylated nucleotides. Enzyme phosphorylase remove 2-phosphate from phosphorylated nucleotide and change them into the monophosphate state. The energy released in this process is used in forming of hydrogen bonds. New strand will form in the presence of magnesium, ATP, TPP and DNA polymerase enzyme. The adjacent nucleotide found attached to nitrogenous base of each template of DNA strands establish phosphodiester bond and get linked to form replicated DNA strand. As replication proceeds, new area of the parent DNA duplets unwind and separate so that replication proceeds rapidly from place of origin towards the end. RNA primer is removed and gap is filled with complementary nucleotide by meaning of DNA polymerase. Since the two strands of DNA run in anti-parallel direction, the two templates provide different end of replication. Thus, the replication over the two template proceeds in opposite direction. One strand with polarity 5' to 3' to 5' form its complementary strand continuously and it is called the leading strand. But replication is discontinuous on the other template with polarity 5' to 3' because DNA polymerase enzyme can add nucleotide in only one direction. So short segments of replicated DNA are formed by DNA polymerase known as Okazaki fragments which are joined together by meaning of DNA ligase enzyme. So this DNA strand built up of Okazaki fragments is called a lagging strand. Proofreading and DNA repair. A wrong base is sometimes introduced during replication. DNA polymerase goes back, removes the wrong base, allows the addition of proper base and then proceeds forward. The newly formed segment is sealed by DNA ligase. Then we have termination of DNA replication. Termination of DNA replication takes place by telomere replication. The end of the eukaryotic linear chromosomes are known as telomere, which have repetitive sequence that do not code for a particular gene. As the enzyme DNA polymerase add nucleotide in only one direction, the synthesis of leading strand is continuous until the end of chromosomes is reached. But there is no place for primer on the lagging strand to be made for the DNA fragment to be copied at the end of the chromosome. So the enzyme telomerase attaches to the end of the chromosome and the complementary bases are added to the RNA template at the end of the DNA strand. After the elongation of lagging strand template, the DNA polymerase enzyme add nucleotides that are complementary to end of the chromosomes. Thus, the end of the chromosomes are replicated. Okay, the summary of DNA replication. It involves 5 steps. Number 1, helicase unwind the parental double helix. Number 2, single strand binding protein stabilizes the unwound parental DNA. Number 3, leading strand is synthesized continuously in 5' to 3' direction by DNA polymerase. Number 4, lagging strand is synthesized discontinuously. Primase synthesize a short RNA primer which is extended by DNA polymerase to form a Okazaki fragment. After the RNA primer is replaced by DNA ligase join the Okazaki fragment to grow to the growing strand, and this overall take place in one direction which is 5' to 3'. The end problem of linear DNA replication. End of the eukaryotic chromosomes consist of telomeres, short sequences repeated hundreds to thousands of times. These repeats provide a buffer against chromosome shortening during the replication. Although most somatic cells shows this chromosome shortening, some cell types do not because they have a telomerase enzyme that adds telomere repeats to the chromosome ends. Then we have repair of errors in DNA. Errors are made in DNA during replication or at any other time during the life of the cell. Three types of repair mechanism operate to correct the error. Number 1 is proofreading, a mechanism for correcting error made by DNA polymerase during the replication. Number 2 is mismatch repair, a mechanism of correcting error made during replication that escape proofreading. Excision repair, mechanism for correcting various kinds of DNA damage such as those caused by a chemical and radiation. Errors in DNA are corrected following three basic steps. Number 1 is recognition of DNA error and its removal. Number 2, replacing the removed DNA by new DNA synthesis using a repair DNA polymerase. Number 3, sealing the new DNA to the old DNA using DNA ligase. Let's look at the proofreading of DNA polymerase. So number 1, polymerization activity of DNA polymerase adds DNA nucleotides to the new chain in 5' to 3' direction using the complementary base pairing rule. Number 2, rarely the DNA polymerase add a mispaired nucleotide. 3. DNA polymerase recognize the mismatched base pairs. The enzyme reverses using the 3' to 5' exonuclease to remove the mispaired nucleotide from the strand. DNA polymerase resume its polymerization activity in forward direction, extending the new chain in 5' to 3' direction. So this is the proofreading method by our DNA polymerase. Then we have repair mismatched base in replicated DNA. Step number 1, mismatched repair protein scan DNA for a mispaired base that was missed in proofreading and cleaves the backbone of new strand on each side of the mismatch. The repair protein removes the several to many bases, including the mismatched base, leaving a gap in the DNA. A repair DNA polymerase fills in the gap with its 5' to 3' polymerizing activity using the template strand as a guide. Then DNA ligase seals the nick left after the gap filling to complete the repair. So this was your proofreading and repair of mismatched base by the DNA polymerase. Then we move on to from DNA to protein. This is your next unit. Defining central dogma of biology. Central dogma is the process in which genetic information flows from DNA to RNA to make a functional product protein. The central dogma illustrates the flow of genetic information in the cell, DNA replication and coding. For RNA through the transcription process and further RNA codes for protein by translation. The concept of a sequence of interaction can be understood through the framework. The most common includes biopolymer. Major category of biopolymers include proteins, RNA and DNA that are further divided into the general transfer, unknown transfer and the special transfer. Special transfers occur in an exceptional cases in lab. General transfer occurs in almost all cells. It describes the regular flow of information through transcription and translation. And unknown transfer are said to be never to occur. Then we have outline of the transcription process. So this is the first step of gene expression where information is passed on from one structure to another. During transcription, RNA molecules are initiated, elongated and then terminated. RNA form is the non-genetic RNA. Transcription occurs as a prerequisite for the translation and occurs when there is a need for a particular gene product at a specific time or a specific tissue. Only one strand of DNA called the template strand is replicated during the transcription and resulting DNA strand are single-stranded messenger RNA. Transcription of a gene occurs near the chromosomal site of the gene, which is usually short segment of the chromosome. Overall process of transcription is highly controlled process, catalyzed and regulated by the enzyme, DNA-dependent RNA polymerase. First step is recognition of specific DNA sequence termed promoter sequence that signify the beginning of the gene. It is then followed by the separation of two strands of the DNA and replication of one strand of the RNA polymerase. RNA polymerase in eukaryotes is different and complex than that in the prokaryotes. Sequence formed after the replication is complementary to the template sequence as the process follows the complementary base pairing rule of the DNA except the thymine is replaced by uracil. In prokaryotes, the overall process is regulated by protein that function as signals or operators and terminate the process of physically blocking the RNA polymerase once the process is complete. In eukaryotes, various proteins termed as transcription factors are involved in the regulation of transcription. Besides, the post-transcriptional modification also take place in eukaryotes, where the mRNA is edited by the process of splicing before the mature mRNA reaches the ribosome for translation. The mRNA thus produced act as a blueprint for the protein synthesis during the process of translation. Depending on the sequence of DNA chosen for transcription, rRNA tRNA synthesis also occurs. Transcription occurs in the nucleus of eukaryotes and cytoplasm of prokaryotes, where the enzymes and the transcriptional factors are available. It is inhibited by some antibiotics like rifamycin and 8-hydroxyquinoline. The process can be detected by the method like RT-PCR, DNA microarray, in-site hybridization, and northern blotting. Then we have translation. It is the second and last step of gene expression where the information encoded on the mRNA sequence result in an amino acid sequence. The starting point of translation is the mRNA formed from the process of transcription of a particular DNA sequence. Thus, translation follows transcription. Even though the information on mRNA is utilized to produce the amino acid sequence, other forms of RNA like tRNA is also required for the process. Like transcription, translation is also controlled by various factors and enzymes, where the most important enzyme is amino acetyl tRNA synthetase. Translation begins with initiation step, which involves the binding of mRNA to the ribosomes, followed by a transfer and binding of activated amino acid to the tRNA molecule. The second step is elongation, where two amino acids are joined by the peptide bond as the mRNA and ribosomes move with respect to one another to ensure the translation of codons successfully. Once all the codons are translated, the resultant polypeptide sequence is dissociated from the translation complex, and the ribosomes are also released to begin another process of translation. Termination is followed by post-translation modification, where the polypeptide needs to be folded to obtain the 3D configuration. This occurs in the endoplasmic reticulum and Golgi apparatus of the cell, and thus polypeptide chain are moved to those organelles. Other modifications are chemical in nature and involve the attachment of functional groups to the peptide sequence. Process of translation is then regulated by binding of ribosomal subunits to the translation complex. Ribosome function as enzyme for the regulation of the various steps. In eukaryotes, translations occur in ribosome associated with endoplasmic reticulum, whereas in prokaryotes it occurs in cytoplasm. The translation is inhibited by antibiotics like tetracycline, chloramphenicol, streptomycin, erythromycin, etc. Similarly, the process of translation can be detected by methods like Western blotting, immunoblotting, enzyme assay, and protein sequencing. Then we have genetic codes. The genetic code comprises the complete information of protein manufactured from the RNA. It is sequence of base pair of amino acid that codes for protein to be synthesized. Thus, a change in the sequence can alter the formation of amino acid. Decoding the genetic code was a real challenge for scientists. Four nitrogenous base and three nucleotides together form a triplet codon which codes for one amino acid. Thus, the number of possible amino acid would be 4 times 4 times 4, which will be 64, but we have only 20 naturally existing amino acids. This was explained by the feature of genetic code, which are as follows. Some amino acids are coded by more than one codon, thus making them degenerate. Each codon codes only for one specific amino acid. The codes are universal irrespective of type of organisms, such as CGU would code for arginine in animal as well as in bacteria, but exceptions exist. Out of 64 codons, 3 are stop codons which do not code for any amino acid and thus end the process of translation. AUG coding of methionine is the only codon that acts as a initiator codon. Difference between exons and introns. Exons are termed nucleic acids coding sequence which are present in mRNA, and introns are the non-coding sequence present in H and RNA, which are removed by RNA splicing before translation. The intron sequence frequently changes with time, whereas the exon sequence are highly conserved. The table shows the difference between exon and introns. Introns are found in eukaryotes only. They are non-coding areas of the DNA. Introns are non-coding part of H and RNA which are removed before translation by RNA splicing to form mRNA. The sequence of intron frequently changes over time. In other words, they are less conserved. DNA base found in between exons. Introns are removed in the nucleus before the mRNA moves to the cytoplasm. Exons, they are found in both prokaryotes and eukaryotes. They are coding area for the DNA. Exons are nucleotide sequence in the mRNA which codes for protein. Exons are highly conserved DNA bases that are translated into protein, and mature mRNA contain exons and moves to the cytoplasm from nucleus. Production of mRNA in eukaryotes. A gene encoding and mRNA molecules include the promoter, which is recognized by regulatory protein and transcription factors that promote DNA unwinding and the initiation of transcription by an RNA polymerase. Transcription in eukaryote produces a pre-mRNA molecule that consists of an 59 cap, the 59 untranslated region, interspersed exon and introns, and the 39 untranslated region and the 39 polytail. The 59 cap and 39 polytail are not encoded in DNA. 59 cap is added by a capping enzymes and the 39 polytail is added by poly-A polymerase once the 39 end of the pre-mRNA is generated by cleavage. Introns in pre-mRNA are removed to produce functional mRNA by splicing. snRNPs binds to the intron loop them out of the pre-mRNA. Clip the intron at each exon boundary and join the adjacent exon together. In the living cell, transcription, pre-mRNA processing, and the nuclear export of finished mRNA are often coupled, resulting in continuous process rather than the separate event. Many pre-mRNAs are subjected to alternative splicing, a process that joins exons in different combinations to produce different mRNA encoded by the same gene. Translation of each mRNA produced in this way generates a protein with different function. Analysis of human transcriptome has shown that there is pervasive transcription of genome and that there is significant amount of potential variation in gene expression for protein encoding gene because of the gene isoforms, the different form of mRNAs that can be transcribed from a gene. Genetic changes that affect the protein structure and function. Genetic changes may lead to a mutation in the genes, which in turn translated into the malfunctioning protein. Transcription is the process by which DNA is converted to mRNA, and translation is the process in which mRNA is converted into protein. Change in the DNA sequence causes mutations. Sudden changes in sequences of DNA may cause a morphological characteristic changes in an organism. An organism that has mutation is termed as mutants. Type of mutation. There are two types of mutation based on their genotype and phenotype. They are point mutation and frame shift mutation. Point mutation, it occurs when nucleotide replaces the specific nucleotide sequence, causing a small change in phenotype of an organism. It is subdivided into two types, namely translation and transversion. Translation, the substitute of an purine with another purine or any pyrimidine with another pyrimidine is termed as translation. Transversion, the substitution of purine by pyrimidine comes under the transversion. Based on its type, it is subdivided into silent mutation, missense mutation, and nonsense mutation. Silent mutation. The silent mutation is also called as a neutral mutation, in which mutated codon codes for the same amino acid as the original codon. Though the amino acid is similar to the original one, it does not affect the structure and function of the protein. Missense mutation. In this mutation, it codes for different amino acid other than the original. Thus, the new amino acid formed is altered, which affects the protein formation. Such protein may be inactive completely or less active. If the changed amino acid still reside in the active side of the protein, it will make it into the non-functional protein. This in turn may cause a phenotypic change in organism. Nonsense mutation, the mutation which changes the codon to stop or terminate the change, is called as a nonsense mutation. It causes an incomplete synthesis, therefore the result may always produce non-functional protein. It brings an extreme change in the phenotype of an organism's then we have frame shift mutation. This may occur due to the addition or deletion of nucleotide in the DNA sequence that may cause changes in the reading frame of mRNA. In mRNA, the three bases form the codon. Addition or deletion may cause a change in entire frame shift. Insertion or deletion in the three base pairs may lead to add or remove a whole codon, which results in change over the single amino acid of the polypeptide chain. Okay, with this we are at the end of the unit. If you have any questions, feel free to message me. Thank you.